Wow, how fun to be back here in Vegas at this meeting. Gosh, so happy to be here. It's such an honor to be on this stage. And I've been here a few times before, and every time I've been here, I've gotten to share stories with you about this forward-thinking, happy, positive discussion about biotechnology and how it will all touch our lives in the very near future. Today, I want to talk to you about how many of these things haven't come to fruition as we planned. And I hope I make you mad. I want to make you angry. I want to motivate you and deputize you to take action to help me bring these things to light. So I'm really excited to be here. That's the mission today. And so I'll show you some of these examples. So where do I come from in all of this? When I've been a scientist my entire life. I grew up in a house of science. And when I was going to kindergarten, I couldn't wait to go, because it was like science fair, science fair, science fair, science fair. And when I got to kindergarten the first day, I said, when do I sign up for science fair? And the teacher said, that's only for the 5th through 8th graders. And I was heartbroken, and I cried till they let me in. And I got to present my work, The Charismatic Megafauna of the Cretaceous Tertiary Period. And a year later, I was going the first day of class to first grade, and this picture shows me and my little sister, her first day of kindergarten. And you could see even then, I had the professor thing kind of nailed down. You know, I had the Dumb and Dumber haircut. I still have the pants. But I've been in science for a long time, and I grew up with genetic engineering. I took the first book of genetic engineering, recombinant DNA technology, out of the Downers Grove Public Library when I was 10 years old. And in 1980s, we saw the benefits of biotech really start to emerge. This picture is, those white things are pancreases. That's not Pee Wee Herman making hot dogs. That's the beginning of the production of insulin, which used to be derived from 10,000 pounds of pancreases to make 1 pound of insulin. With biotechnology, we were able to take the human gene for insulin, put that into bacteria, and now have bacteria that were reprogrammed genetically to make 100% human insulin. Now, if that's not cool enough, in the 80s, we also had enzymes that would make cheese. That we used to have to get the enzyme purified from the guts of ruminants. That now we were able to take the gene from actually a camel, put it into a fungus, culture large vats of this, and make enzymes that were responsible for causing the curdling of milk to make cheese. Other technology we didn't mind so much. I mean, here's Cologuard. I mean, this uses a biotechnology test to tell if you have precursors of colorectal cancers. First time I ever crapped in a box without anger in my heart. We also have the 23andMe kits, where you can get all your genes analyzed and tell about predispositions for different diseases. You also can learn about how your family's been lying to you about your ancestry. You also can meet thousands of relatives, distant relatives that you never wanted to know, who now are excited about meeting you. So all of these are applications that we were really excited about. One of the more recent breakthroughs was the COVID vaccine that many of us thought was amazing. We went from the genetic sequence of a new virus to a vaccine in 42 days. It went into that first arm. Amazing stuff. Yet we still saw a lot of social pushback. And while scientists agree this saved millions of lives, there still are people out there who haven't accepted it, who won't accept it. And if the next pandemic comes, will not accept, or I should say when the next pandemic comes, will not accept the mRNA-based therapies that can save their lives. I mean, this is a real problem. People get excited about innovation, at least some of the time. And so this is what we're going to talk about today, is how we, and I mean we, can grease the skids of communication to make innovation more palatable. How do we have those difficult conversations in social media space where the conversations are taking place? So here's a crash course in genetic engineering. For those of you who haven't been doing this for a long time, think of it like this. You've got parts of our cells are a lot like parts of a computer. The central dogma of molecular biology says the master blueprint is DNA, a lot like the hard drive on your computer. It's tucked away, it's working hard, you never want to see it, it's doing its thing. Transient pieces of that information can be put onto a intermediate step. in this case a USB drive, which is the RNA. The RNA is a small piece of the genetic information from the hard drive that now goes out into the cell. Then finally, you have the printer that's there, which is the protein production. The proteins that are the catalysts, the catalytic and structural units of the cell. They're the things that do the work. So the information is in the nucleus. That then goes through this intermediate and gives us some sort of a product. In the case of the COVID vaccine, we used the power of that USB drive. We used the intermediate RNA species to produce some sort of product. We introduced the RNA into your cell of your arm, which then made a protein that then the body, here in this case the S protein of the SARS-CoV-2 virus, that now your body would use to mount an antigenic response if the virus actually came along. It kind of was a wanted poster. It showed your body what it needed to look out for. And this is why it was such a wonderful approach that now is same approach is being used to cure cancer. Pancreatic ductal cancers were previously a death sentence, very low survival rate for more than 5 years. New therapies coming out this year with a vaccination against aberrant proteins from those cancers mean that 50% of the people in those trials went into remission. This is great stuff. So excited. Here's one for science. Wow, science gets applause in this room. I love it. Whew. A lot better than when I do it on the street corner. So how do we put genes into a cell? How do we make genetic changes in an organism or cell? And it's the same way we do it in the library. You might go and put a new book on the shelf, new information into the library, just like a new gene into the genes of the nucleus of the cell. You might do what we do in Florida and get rid of some of that information that's there. The other alternative is the modern approach where we can go into a book, identify specific letters, and remove them and completely flip the meaning of the sentence. Part A. Good times. So it's ability for us to make very precise genetic manipulations that allow us to make powerful changes inside cells that then can pass on those traits to the next generation or solve an immediate problem. This is how genetic engineering has been so revolutionary. The first application I'll show you is how we were able to protect livestock. And really cool new innovations that have changed the way that we farm. There's a number of threats to farming that are really important, that really do dictate not just the cost of our food, but also animal welfare. Some of the major ones are avian influenza. That millions of birds are bulldozed every single year because they contract the disease. One bird on the farm gets avian influenza, all of them must be destroyed. We've had a transgenic chicken, a new book on the shelf in the chicken that cannot pass the disease. It gets infected, it can't spread it. That we've had since 2011, not used. We have an mRNA-based vaccine since 2020, not used. We have solutions in pigs, we have solution in cattle. The best ones in cattle that don't grow horns. Much safer for the animals, much safer for the handlers. We have cattle where 75% of the offspring are male. Now, why would that be important? Well, males have better feed conversion. For every pound of food, you get more pounds of meat out of a male than a female. So having 75% males is a sustainable alternative. Yet all of these technologies, which have been tested, which work great, not used. Mostly because of export issues. Other countries will not accept them from us if we use those technologies. So the animals suffer. The misinformation ecosystem online is primarily, you got it, didn't you? I heard somebody laugh. Yeah, I'm mRNA free. Find me a cow that has no mRNA and I'll show you a cow that doesn't exist, right? So the misinformation ecosystem is extreme, and it's amazing how bad it is, that this is the kind of pushback we get against good technology that keeps it from being adopted. Biofortification is a major one for me. How do we make plants that can help people? And this is an old story, but I want to reintroduce you to it. Vitamin A deficiency is a problem worldwide. And in the countries where they're experiencing the most blindness from vitamin A deficiency correlate with where the food has the lowest amount of vitamin A. leads to loss of night vision, and eventual blindness, and eventually death. And the reason this happens is because the orange pigments that we enjoy in our crops aren't present in most world food staples. So the basis of the diet is devoid of most beta-carotene, the orange stuff in carrots. So scientists had this great idea back in the 90s to put this stuff back into, put beta-carotene producing enzymes into the world food staples. Put a couple of new books on the shelf that gives that crop the ability to produce the orange stuff. And they did it in the 1990s. They did it in rice primarily, which is 40% of the world's calories. And there it is, golden rice, great stuff. It was approved and grown and eventually approved in the Philippines for use in Philippine populations where vitamin A is still a problem. The matoke banana. In the lakes region of Africa, this is a food staple. There is not a meal that you eat. If it doesn't have matoke, it's not a meal. And that's what they would say there. There's also bacterial wilt diseases that threaten smallholder farmers. I had the opportunity to visit farms and spend time at the Kiwanda Research Park outside of Kampala. And there I met Dr. Priver Nemanya. And Dr. Nemanya, she wanted to solve a problem. She's a Ugandan. She wanted to solve a problem for the people of her country. And she generated the beta-carotene banana. And you can see a little orange cast to that banana. And the matoke banana that has vitamin A, she created this thing. And it's behind a barbed wire fence, a guy with a gun outside, and you have to sign in and sign out before you go into that field. And this is 2017. You stand under these beautiful trees that can solve problems for farmers on the other side of that barbed wire fence. I got to go there because I was to speak with the science minister, who was a medical doctor who didn't know much about genetic engineering or crops, and speak to him before he spoke to Parliament, because he was going to try to get a framework to begin studying genetic engineering and its risks. He was going to Parliament to set the stage for approval of that solution that Dr. Nemanya made. A few weeks later, I got a text in the mail that it had been approved. I read it on my phone and I cried. Don't applaud yet, because a few weeks later after that, the president vetoed it. So here's a solution made by Ugandans for Ugandans that gives the stuff in carrots to solve a problem of blindness in the people of Uganda. Yet misinformation is what stopped it from moving forward. The radio stations would have ads about how it would cause cancer. And these were all sponsored by friendly organizations in the West, NGOs like Greenpeace. Unbelievable that this would happen. Golden rice was approved in 2021, and it was now defeated in 2024. And when The Guardian comes out and says that it's a catastrophe, you know it is. I mean, this is a publication that's been against genetic engineering for a long time. And in this case, they point out that this is actually something that's deleterious. The world's most deadly animal is not the shark. It's the mosquito. And a mosquito kills many people because of malaria, an easily transmittable disease, along with many other types of easily insect vector diseases. And a solution was proposed some time ago now to stop the spread of these diseases by an ingenious technology. And the idea was that there are genes that are lethal to the larval stage of the insect. So as the insect goes from its egg to a larval form, the larvae then matures into then moves to a mature form of the insect. If you could stop the larvae from growing, you never get a mosquito. So they added a lethal gene that would only be expressed inside the larval form, but you could turn that gene off with tetracycline, just an average laboratory antibiotic. So you grow them in the vat in the lab in the presence of tetracycline, you shut off that gene, you let them progress to be adults, and now you release them. And now those adults go out and mate with the population, and the next generation of larvae, no tetracycline, no mosquito maturation. And numbers crashed. They didn't go to zero. And these are invasive species in most places. They're not messing with native ecosystems. If anything, they're making them better. They use similar strategies to combat diamondback moth and fall armyworm, two insect pests that have massive agricultural losses. However, these two technologies too have pushback and are not being deployed in the places where they're most needed. mostly because of activism from the West that has tarnished the reputation of these good technologies. The last thing I'll talk about in terms of technologies is a new one that's just fantastic. And this is ending a painful, debilitating disease of sickle cell disease. Sickle cell disease affects approximately 100,000 African Americans, mostly those of more recent African derivation, and affects the joints, affects small blood vessel organs, affects the extremities, can be extremely painful and cause premature death. It's caused by and this is, well, we'll talk about her in a minute. The basis of this is a single change in the library of the genome, a single change in one letter that causes the cell to take on a sickle cell shape, or sickle shape. What if we could correct that? What if technology allowed you to change that letter back to the correct letter and then reinstall that? And that's what happened with Victoria Gray. She's the Neil Armstrong of gene therapies. What she had done was the wrong copy of the gene was corrected in the lab. They took out her stem cells, corrected the sequence in the stem cell, and then put that back into her bone marrow. There it repopulated only with the correct form of the bone marrow. And the correct form of the bone marrow makes all the other blood cells. And her sickle cell disease, oops, where'd my slide there? Oh, there it is. They put this back into her, and her sickle cell disease was essentially cured. This is amazing stuff. And I think this should be, if it was up to me, this should be something that our government or somebody should be making available to everybody who needs it. In the past, we heard earlier about Tuskegee. We know that clinical trials in African Americans are not done the same way with tests for drug efficacy that they are for others. This would be a great way to kind of start to level that playing field a little bit. Today she's fine, and I'd love to see this technology get out to more people. However, because of the skepticism from Tuskegee and other types of atrocities that have been committed, the communities are very wary about accepting this technology. And there needs to be more work in developing the social license to ensure that anyone who wants this technology can get it. The other big problem is the cost. It costs companies billions of dollars to come up with this therapy that now they only have to deliver one time. It's a one-time shot and probably a cure. Hard to know how it's going to work in the long term, but should be durable. And so how do you manage this from a public health standpoint, that you can correct the problem? The companies that made it maybe won't profit like they wanted to. There's many very strange edges to this particular problem, but we really need to find a way to make it happen. The bottom line is that I hope I've convinced you that we don't have an innovation problem. We have a problem. We're able to do a lot of cool stuff, and I didn't talk about a lot of things that I really wanted to talk about. I was feverishly ripping slides out of a presentation this morning. Lots of good things that we can do. The problem is we have a communication problem. Scientists don't know how to talk to people. That's what I trained them to do. But still, we're not very good at it. The problem is that the bad guys are really good at talking to people. The Dirty Dozen comes up every year and convinces people that their food is poison, that there's presence of extreme amounts of pesticides. The reason they know there's anything there is because they're using USDA data that show food is safe and that the levels are low. Yet for them, saying that something is present is enough to say it's not zero, so it's dangerous. In front of the Senate last month, RFK Jr., the food babe, and a bunch of internet influencers got time in front of the Senate to convince them that their food was poisoning them and that their food is a problem. These aren't scientists, these aren't experts, these are folks trying to sell supplements and use the internet to gain followers. They have a full-time job of producing disinformation that's going to be affecting food and farming. And they're gaining momentum. RFKJ, he could very well have a part in the next administration if that comes to fruition. I mean, it's scary times, folks. The internet is still awful. Bioengineered food awareness is 1 million followers altogether, orchestrated out of Morocco, and is nothing but disinformation and risk about food and farming. So how do we fix it? We need to control the narrative. We need to do it as scientists in the scientific enthusiast community. We need to get in and do that. And so how do we do it? The bottom line is we need to stop dealing with an emotional problem with facts. And we heard Dr. Novella earlier say that, this is one place where facts can matter, and they do, but only after you've earned trust. You can't change people's mind with an avalanche of information. You have to build trust. And what is trust? Trust comes from a combination of competence, reliability, and intimacy over self-motivation. So we're all pretty competent as scientists and reliable, but scientists in general lack intimacy. They don't show they care. There's also the question of why they do what they do. Are you doing it because you're owned by Monsanto or because you genuinely care? In social media, we're not managing those perceptions properly. So we need to be working on the do we care? And we can do that by connecting through common values. When we talk about this as scientists, that we're interested because we want to feed more people. We want farmers to be in business. We want ecological sustainability and safe foods for consumers. Now we start to connect with the audiences who are usually repelled from us. This is how we manage those conversations in social media, because that's where this is all gained and lost. We have to engage properly, and I won't go through this whole slide except for 2 parts. The 3 most important words in the human language to change somebody's mind is help me understand. Listen to somebody's concerns. Help them articulate what their concerns are. Understand where they're coming from, even if you disagree with them. Don't listen to debate, listen to understand. Because from that standpoint, you're developing a sense of empathy. You're showing them that you care enough, intimacy, to be able to listen to them and understand and validate their concerns. This is so important. The other line on this table that makes a lot of sense, I really like, is you don't treat people like their opponents. I did for a long time. What's wrong with you, you idiot? It doesn't change hearts and minds. If anything, you treat them like victims. These are folks who have been lied to by bad people. They're people who have been misinformed, disinformed, people who have been fed false information, sold supplements, you name it. And they're victims, and we need to treat them that way and treat them softly. And remember, the other thing is that your audience in social media isn't the person who's difficult. They're not the troll. It's the people watching you interact with the troll. And you can earn trust by taking the high road and being the kind heart in that room. The most important thing that this room can do is amplify the information of others. Now, we all know that you can create content, podcasts or whatever, and share that with 1,000 people. Pretty easy. But it's the people who amplify that and share it with another 1,000 people that now take it to 1 million. So as you're sitting in front of your computer and you're on Twitter or X, whatever it is, Facebook, whatever your major place is, and you find an article that's good, don't share it because you like it or you think it's cute or funny or whatever. Share it because somebody in your network needs to see it. Share it because it's great information by someone who cares that you can share to convince others in your network how important it is, that then they may share with their networks. This is how we get ahead in this discussion. You amplify the experts, you know a few trusted sources, and you constantly engage. This is how we are going to change the acceptance of good technologies. And it's critical that we do. They're already changing lives. They're changing things in the industrialized world as well as the developing world. And as we continue to engage, the people who are against these technologies, there's only a few of them. There's an awful lot of us. And if we all take the time to share the information from the experts, that's how we change this narrative. I'll conclude by talking about my friend Vance Crowe. Vance, if you don't know Vance, he does a wonderful podcast. He's a great speaker. Vance also was in I'll share a story of his. He also was in the Peace Corps. He was in Kenya, in a region of Kenya, to talk about malaria because the roads were coming to that area. And with the roads would come mosquitoes. And Vance was teaching people about mosquito netting and about repellents and ways that you can manage your exposure to limit the possibility of malaria. And he was in this town center where people would go after work at the end of the day, and they would share different food, trade food, do different stuff out together in the center of this village. And a car pulled up, a beautiful car that was using the new road. And a man came out in a beautiful suit, and he put a box down on the ground, and he stood on the box, and he said, "Ladies and gentlemen, gather round. Let us pray." And everyone did. And he said, I've heard of the new disease that's coming to the area. So I went down by the river and I met a man, and he was a holy man. And I looked into his eyes and I saw he was pure. And he told me that he had blessed water. That if you took a little drop of this blessed water and washed with it, you'd be protected from the disease. Now I have vials of this water I can sell to you for only 3 days' wages. Who wants one? And Vance said, these are my people. I've been talking to them. I'm part of the town. They understand me. You know, they're all for it. They're going to tell this guy to get lost. Everybody ran up with money in hand to get a bottle of that water. How many people died because of a charlatan who was making money off of people's fear of a disease? Vance at that point said, I will dedicate the rest of my life to knocking the man off the box. And I realized when I heard that story from Vance that that's what I've been trying to do, is knock that man off the box. How do we go out and deal with the disinformation and the purveyors that are profiting off of false information that harms others? So really what I'm asking you to do is help me knock the man off the box. You may knock off somebody who could benefit, who you may take steps that could ultimately benefit someone in your family, someone you love, maybe a child in the developing world. And so keep that in mind as you engage in social media. It's all about how we're going to knock that man off the box. Thank you so much for your time. This was wonderful. Thank you.